We're a technology development lab focused on trying to make the best types of measurements and the best types of software for analyzing those measurements using a mass spectrometer. But we're interested in kind of what's going on in the tumor microenvironment from an immune cell perspective. One of the problems with this type of experiment is that there are not going to be that many copies of each cell in that specific tumor. So before using the Stellar mass spectrometer, we were using an Exploris 480 for this type of experiment. And this is an extremely specific instrument that's able to make very precise measurements with very high mass accuracy, with high dynamic range. But we have to take a lot longer to make any individual measurement, and we have to use a lot more sample in those particular cases.
We have to do injections, which would be a single DIA injection, using around 40 to 50,000 T cells. And so with using that many cells, we have been forced to really scale up to large experiments that are both costly in terms of time, resources, and also instrument time as well. And
our goal for this was to see, is it possible to dial back the amount of material that we need to make a good quantitative measurement in a similar throughput way or a faster throughput way onto the Stellar mass spectrometer, and tried to see how much of the workflow we could actually do on the Stellar mass spectrometer alone. And we were surprised by how good quality libraries we could actually generate using just the ion trap on its own. The advantage of trying to do a data independent acquisition based library is that we can also do it without additional chemical fractionation. And this cuts out so many steps in our workflow for trying to make the best measurements possible. Because our lab is focused around trying to make measurements on thermoscientific mass spectrometers, we have designed our software to actually output an inclusion list directly for using on the Stellar mass spectrometer. And so the software automatically generates all of the way through from target refinement and selection out to an inclusion list that we can plug right into the method editor so that we can make our measurements directly with ease. And so we took a series of isolated T cells and tried to see how good we could make measurements using gas phase fractionation for library generation, as well as trying to do our targeted experiments on as low input as possible. And our focus with this was trying to see within a matrix matched calibration curve, how low could we actually make quantitative measurements for these particular peptides using transitions that we've refined using our software platform. And so we started within a background of 100 nanograms. How low could we measure our key proteins of interest? And then at 10 nanograms. And we were shocked at how accurate and how quantitative our data was at 10 nanograms. So we went further. We went down to 1 nanogram. Now with 1 nanogram, we're talking about 3 to 4 to 5 cells in total for a standard cell line.
We were pretty surprised by the aspect that we could still see quantitative peptides down at the 1 nanogram level. We were very happy with the results.
And then we were able to measure down to 10 picograms, so 2 orders of magnitude of quantitative range, which gave us the confidence to be able to say we could make measurements of T cells with a very little starting material.
One of the most exciting parts of this is that all of the data collection that happened for the paper was within 2 weeks. So we were able to optimize both our DIA methods, run our libraries, optimize our PRM methods, and run those PRMs to the point where we're able to run multiple calibration curves. And we probably, if we would have really wanted to, we could have built multiple targeted assays, but we were really just interested in a specific aspect of T-cell biology, but we were definitely able to do the entire workflow within 2 weeks, including the optimization.